I'm Ann Walter in the biology department, and this is a case study that I developed with Becky Sanft and have taught with Minnie.
And I'm Becky Sanft. I'm at UNC Ashville and happy to be here.
Great. And we're going to take you through a lesson. So, as if you were in the last session, you realize that this case study occurs towards the end of an undergraduate mathematical modeling course. In this particular case study, where we're looking at vaccination response on the consequences of a tumor growth, as diagrammed in this very detailed model right here. A vaccine is given, immune system does something, and hopefully the tumor will be get smaller due to the effect of the immune system. So the goal of this case study is really to, first of all, understand why modeling is important in medicine. And in particular, a case like this where microenvironments, i.e. the immune system, and the really specific spot of the tumor are inaccessible. We can't get data from them. So how can we learn anything if we only have the final output data, the size of the tumor? So to carry on, we first have to describe this system by formulating the differential equations that can tell us something about the tumor, the vaccine cells, and the immune response. And the prostate cancer cells in this particular model. Next. The data that we have is PSA. PSA is a substance that can be measured in the blood. It has direct relationship to this number of prostate cells that are in the body. So we can use PSA to estimate the size of the particular tumor. But that's the data we have. The strategy for the therapy is to give a vaccination, and the vaccine is it's essentially dead prostate cells. So will cause an immune response. We give vaccination on a schedule. So how much vaccine, what is the vaccine schedule, and will that actually stabilize or decrease the number of tumor cells? We use sensitivity analysis to decide whether or not the parameters that we're using in our model are sensitive or not, which ones we should pay attention to, and which ones might help us decrease the number of tumor cells. So what happens right at the beginning? We have several compartments in our model. Our first compartment is injecting a vaccine. Orange vaccine gets injected and activates some dendritic cells that then migrate out of the skin where the vaccine is injected to where they're going to activate the immune response. So our first job is going to be to figure out, guess what's going to happen when we inject the vaccine cells at time 0, and figure out what do you think is going to happen to the number of vaccine cells. They do disappear into the dendritic cells. And then what's going to happen to the dendritic cells over time. So we're going to invite you to do an exercise. So you want to be thinking, we're going to open up some Jamboards for you.
Yeah, so in the chat, there is a link to a Jamboard. So we're having us all be students here, how we would do this in a classroom. So click on that link to the Jamboard, and pages 1 and 2 have some axes there, and you can sketch.
Right. So go ahead, feel free. We're not going to grade you. Is everyone able to open the Jamboard?
Yes.
And I'll show it here too. We can see it.
Okay, we're not seeing a lot of activity. Oh, something's happening. Do people know how to use Jamboard?
No.
You can
Oh.
use this pen over here. Do you see this pen? So if I click on that pen, I can draw. I'll erase that. You can even pick your color. So I might draw some curve here, what I think is happening.
Right, so what are you thinking if we inject a vaccine? And we're going to inject a vaccine at sometime. Perfect, all good. And then the dendritic cells activated, they're going to go up. Okay, good. Okay. Ah, okay. Looking good. Okay. Oh, so there's a big question. Are we going to get a plateau or are things going to go up and down? Oh, we have excitement. What do you think? Should we go back? See what's...
All right, let me go back here.
Yeah. All right. So you all have been thinking we inject some vaccine, might go up, then it's going to disappear into these clouds, so it might go down. These dendritic cells get activated, that activation might go up, and then they leave. Let's see what happens. Becky, can we go look?
Yeah. So we've thought about what the curves might look like. So how would we represent these things mathematically, right? So we have the vaccine cells that are being taken up by these dendritic cells. And so we have this NV. So that's the number of vaccine cells required to induce this maturation of one dendritic cell. Ki would be the rate at which that vaccine stimulates for these naive cells to start maturing. And for the DMs here, those dendritic cells, right, there's that rate of them becoming those DMs. And then we are assuming here that these DMs migrate from the skin into the lymph node, and that rate's linearly proportional to the number of DMs here. And so based on those drawings that you made, we formulated a model. We can solve this set of equations and see, does it match our intuition of what we drew? And so this is an example of, now remember, this case study is toward the end, right, of the semester, what they've learned. And so by this point, they can solve differential equations numerically, they can make sliders. And so here, if you just look at this first part, that's one injection of vaccine cells. And so some of you kind of drew, I think, the vaccine decaying. It's decaying really quickly here on this scale here. And then let's see, so that the DMs take up the vaccine, they're gonna start maturing. So we get the DMs here. And we can play around with this. So notice the vaccine, right? Those are taken up very quickly. And so we can think about schedules of injections, right? So this is assuming an injection interval. So every 30 days, we're injecting vaccine cells, and this is the number of vaccine cells. And so we can vary these things. We can change the interval, right? And see how that changes. We can change the number of vaccine cells, right, that are injected. But what we notice here is that that vaccine is taken up really quickly. So we see the importance of these injection intervals. And so the next question is, this was just the initial, like what happens, right? When you inject those vaccine cells, and there's a whole cascade of events, right? So how do these parameters here, how do these affect the number of tumor cells over time?
And here we've got the mess. So in this slide, we're seeing the first part that we saw, the injection of the vaccine, the activation of the dendritic cells that start them to migrate. And once they've migrated into the lymph node, they turn green because they are now in a different space. And they're in a position of being able to activate it, the cytolytic cells that are going to then go to the tumor and make the tumor break. So we see this tumor turning into a star here, breaking. But in the lymph node are a lot of things going on, and this is just a few of them. The dendritic cells wear out. They get tired for a variety of reasons, and they turn into regulatory cells and then to Tregs, which are the blue ones. And those Tregs will ultimately inhibit the cytotoxic cells, the ones that are going to kill the tumor. So we have a lot of things going on. For now, we're going to consider that lymph node, the black box, the same black box that you saw at the very, very beginning of the slide where we had inject a vaccine, stop a tumor, and ask the question, what happens to the tumor? And remember, we can measure the tumor size because we can take a blood draw and we can measure this chemical PSA, and that's going to tell us about the tumor size. That's the only thing that we can measure. We can control the number of cells in the vaccine, the rate of the vaccine, and we can measure the tumor. So we have some more things to think about. So assume we give no treatment. So no vaccine is injected. What do you think the tumor cells are likely to do? Is the tumor going to grow over time? What kind of growth might you expect? And we're using P for tumor cells, prostate tumor. So your first exercise coming next on a Jamboard will be the size of the tumor versus time. And then what do you think is going to happen if the vaccine is injected once a month? Do you think the tumor will go down? How will it behave? So Becky, can we?
Yeah, so it's that same link in the chat, and I think someone might have just joined, so I'll put it there again. But if you go to that same link and just go to page 3, you can start just sketching away.
Oh, good. That's great. We've got axes labeled. I love it. That's the beginning. Okay.
And there is another page too, if anyone would like to do another one.
Yeah. So we're seeing the tumor cells, and we've got 2 models for growth here. Got a nice logarithmic model, got a logistic model. Whoa. Oh, no vaccine. Got it.
Let's see.
You guys are great.
Good. This is fun. All right, that might be.
Okay.
Let's
So
see, let me go back here.
So is anyone ready to think about what would happen if we injected vaccine? Do we do that?
right, well, let's
We'll just
talk about this.
Well, we'll think about it. So presumably the vaccine was going to do something.
Mm-hmm. All right, so at this point, you know, we've thought about these relationships very briefly. I noticed it looked like one curve had the number of tumor cells looks like growing exponentially, right? So that would be this first piece here, right? And then we can talk about that in class, right? That we've thought about logistic growth. So this is an assumption that we're making. So it might not be a good assumption if we're looking over long term. And then we need to have some interaction with those CTLs here. So this first part, oh, and I should go ahead and go here. So the R is that proliferation rate of the tumor. Now the second term, again, is that interaction with those CTLs. And so this looks like a mass action term. We've seen that a lot throughout the semester by the time we do this case study. AP is a maximal tumor killing efficacy by the CTLs. However, it's known that the tumor killing efficacy, it can decrease as the tumor gets larger. And so this acts as a damping term. And so this HP, the higher it is, the more damping we have as the tumor gets bigger. All right, so we've drawn some pictures. We have a model. Again, we're kind of skipping over that black box, that middle part. But when we solve this system of equations, we can plot P over time, right? Because again, that's we see the data on this PSA data. And so this is what we can compare the data to. And so we can look at P over time. And so here we have the number of vaccine cells injected. This is the injection interval. So right now we have no vaccine, and you can see on that model, right, that we're growing exponentially. Now, once we add some vaccine, we start seeing some wiggles, right? And so in that previous graph that we saw, you saw that the vaccine, it was taken up very quickly, right? And those DMs increase and then decrease. So we see every time we get an injection here, we see a little decrease, right? But then, right, on the scale here, the tumor keeps increasing. And so we can use this model thinking about different vaccination strategies, right? So if I increase the dose here, we can get to a level where maybe we're just kind of maintaining a low level of the number of tumor cells. Or if we increase, if we do injections more frequently, maybe we could even decrease. Now, obviously, we talk about kind of the effects of that on a patient. But they can use this model and start thinking about different schedules for these vaccinations. Now, in the beginning, we have students just play with the model, right? We give them some parameter values, but then I'm just going to talk really briefly about this, but they do some parameter estimation. So we have data for different patients, PSA data, and we have to convert that to number of tumor cells. So a data set might be like the points that you see here. Most of the parameter values we can get from different in vitro and in vivo studies. The ones that are tricky to get that tend to be more patient specific is that proliferation rate R. And it tends to be very patient specific on kind of the intensity of the immune response. And so AP can vary quite a lot. And so the students will take a data set here, the pre-vaccine data, they'll use that to get their R, and then the post-vaccine or the vaccine treatment, when that begins, they can do some least squares and use an optimization package in R to find the value of AP that gives the best fit between the model and the data.
Exactly. So now we have two really critical parameters that are very patient specific. But we want to learn something more about what's happening in the middle. As Becky said, we can describe the interactions between these dendritic cells and the creation of the cytolytic T cells, how fast they turn into regulatory cells, which are going to tamp down the system. We have parameters from the literature to fit this sort of internal model, the black box that we talked about before. But one of the questions that we might want to ask is, how important is it that we actually nail those parameter values? We pulled them from the literature. How do we know that they're important, how important they are? How do we know whether they might have anything to do with helping us get rid of the tumor? Maybe we can identify one of these values, and by knowing something about the immune system, make a proposal for how we might improve the tumor. Or not improve the tumor, improve the disappearance of the tumor. So by knowing something about this internal relationship and doing sensitivity analysis, we can begin to think more broadly about the system that we're looking at and how we might proceed if we were recommending to our collaborator in a medical setting. Oh, let me go back. I totally forgot my most important thing. So as you're looking at this, you can see that the blue turns to the green causes orange, the dendritic cells to go up, and the green also gives us the killer cells. The orange cells give us light blue, which turn the killer cells so they go away. If you are looking at these parameters, and they might be hard to see, but you could guess which ones, you might think about or pick one that you would want to either increase or decrease so that maybe the tumor is more likely to go away. So as you look at this model, if we were to increase the rate of regulatory cells, we would guess that we would end up decreasing the number of cytolytic cells and thus have a less effective tumor disappearance. Conversely, if we increase the rate of conversion to cytolytic cells, everything would go away faster. So you might take a look at this series of equations and pick a parameter to look at. And Becky, we can
and then we can quantify the effect. And that's where the sensitivity analysis comes in. So for this exercise, we have students increase each parameter. We have a lot of them in this model. They increase each one by 1% and look at the percent change in number of tumor cells after 30 days. And so we can see which ones have the greatest effect and which have the least effect. So for example, Ki, it seems like the model output is not very sensitive to small changes in Ki. So from a research design perspective, we may not want to put a lot of energy in getting that parameter more accurate. We also see and also this is just a nice model validation tool, right? So students can look at this and see, okay, if I increase this parameter, does it make sense that the change would be positive, or does it make sense that the change would be negative? So this is a nice kind of validating the results. It helps students do that. We see that it seems to be really sensitive to changes in R and AP. So we can look at that a bit more, and Anne will talk about that next.
Great. So we notice that the large range of values of tumor growth rate and killing efficacy, AP, have been found among patients. So one of the things we want students to think about as a result of this sensitivity analysis is whether we could give a single recommendation for a clinical procedure, or whether the results of this sensitivity analysis says that it's really important to model each patient's data and develop a patient by patient individualized medicine, if you will, vaccination regime. So that's an exercise that we have students thinking about. And so then at the very end, we ask students to do what we were asking you to think about, which was to look at each of these parameters, whether they cause a bigger tumor at the end or a smaller tumor at the end, and come up with some kind of suggestion for adjusting the value of that parameter. And this is where some of the biology comes in. Almost all of these parameters are adjustable. It also, maybe they'll go up or down in different ways, but the idea is to be able to use the sensitivity analysis to actually prepare a report on the potential of either stabilizing or decreasing the cancer, given the particular parameter that they're looking at. And some of them, like I say, may or may not be able to be adjusted a lot, but at least it gives them something to think about. What's the literature you should look at? What's the experimental design you might look at next?
So this case study is near the end of our textbook. So thought we would cover this one because it gives you an idea of what students can do by the end of the semester. The model and data used for this case study was taken from this paper. And we also put a couple of links here. There's also a paper kind of going over what we talked about in our first session, just an overview of the St. Olaf College class in particular, and how we've incorporated a Wetlab there. And then Anne is at St. Olaf College. And Anne, I included this here too. I don't know if you want to mention anything about that. I guess that came up during our break, but they have a concentration. It's an interdisciplinary minor in MathBio. So our course that we developed there is really the core course of that concentration.
Exactly.
So thank you. And we would love to hear any questions that you might have.
Hey, let's thank the speakers for the presentation, and then any questions for the speakers? All right.
Hi, sorry, I have a question.
Okay, go ahead, Olaf.
Yes, my name is Myla.
Myla,
Could you
Myla.
please show slide number 15 again? The one that has the systems of equations.
Oops, I'm sorry. I shut that down.
So it looks like we have three. So we have three different areas. And then in each area, we have in each area, we have a system of differential equations. Like in the lymph node, for example, the one in the middle, there are 4 differential equations. Is there a link between these 3 systems? I mean, so individually, I see in the first system, I see V and dm. In the middle one, I see the 4 functions of t. And in the third one, I see p of t.
Right.
Yeah, you can see that we looked at these first 2 equations first because these are decoupled from everything else.
Well, in some ways, but then they link, of course.
Right. So the dm, right. Yeah, go ahead, Anne.
I was just going to say, so
yes, I see it now. I see the dm now in the second. Okay.
Right. And then that allowed this collection of cells either can go to the regulatory cells, which is happening here. You'll see that. Or they will also end up contributing to the cytotoxic cells, DC. And then the regulatory cells are going to start disappearing, the DCs, which is here.
Okay. Yes, you're right. They are
Yeah,
coupled.
no, it's a lot of equations with little subunits, and then it's the C's,
Yes.
are over there killing the peas
Yes.
at a
Thank you.
particular rate. Yeah,
Yeah, good question that we really went quickly through that. So.
yeah.
All right.
I
I
have a
one
question.
Oh,
I
you
have a
have
question.
Arti, go ahead.
So the question is, I liked it. Thank you. It's a very nice, neat presentation. So my question is, are you thinking of going towards partial differential equation when you are thinking of growing? So are you going to increase your X or something like space into these equations, PDE?
So this is for an undergraduate course in math modeling. And so we just don't have time to get into spatial modeling. That would kind of be a next course.
No, I'm not talking about the course, but from the research point of view, because you have done so much good work that if you would like to continue and extend it to PDE so that it will be like a complete model.
Yeah, and actually this was a published model in data here. So we actually did not develop this particular model.
Right.
Oh, okay. Okay.
Right. In
Thank
fact,
you. Mhm.
But yes, you're right. The spatial component gets important.
it does.
When the tumor is growing, so it is growing in space, so it's natural. So.
No, that's absolutely clear. And one of the things that we find ourselves doing a lot of times is having to explain to students, because they figure this out too, is that, for example, the rate constant we've used for moving the activated dendritic cells to the lymph node includes the maturation, so what the biology of the cell is doing, and the movement. So we're hiding some things in these parameters, or I should say chronic et al. We're hiding some things in those parameters so that it's not a complete model, but it's a sufficiently complicated model.
Yes.
There are also a lot of things in the lymph node they are completely living out and we are leaving out. And those are turning up in some of the patient-specific data in the effectiveness. So on the tumor, there's a lot of stuff hiding in that particular parameter.
So you really applied to the patient data and verified what the model is doing?
I'm sorry, can you say that again?
So did you verify your result with the patient-specific data?
So we did do the parameter fitting. The students go through that. Mm-hmm.
Okay, thank you.
Yeah,
Very
but you're right.
neat.
These questions about the spatial component, that's what's fun about this course, because we can kind of ask those questions. How would you do that? And in this course, they're learning such a breadth of tools, right? They're learning a little bit of stats, a little bit of math, a little bit of programming. It really gets them to want to take more courses in these different areas. So it's fun to get those conversations going.
Yeah, I agree with you.
So any other questions for the speakers? Okay, I have a quick question. So about the sensitivity analysis, you guys showed a graph on the sensitivity analysis. Does the student generate those graphs using the programming or, okay.
Mm-hmm. Yeah, and actually those aren't too hard for them. If they know how to solve the set of differential equations, then they can change each parameter. We try to get them to be a little savvy about how they do that when they program it. But yeah, they can create those